testing. Okay, now so now is already 8 o'clock. Okay, so we will start our class now. Okay, so as you guys information I already mentioned in group and also post the channel that starting this week our class will be change date and increase one more day. Will be on Tuesday and Wednesday at 8 o'clock. So starting this week we will have 2-day class and then the day is changed to Monday, from Monday to Tuesday, and then extra one day one Wednesday. So okay, so today and also tomorrow. So tomorrow you also have the class at the same time 8 o'clock too. Okay, so last week we already finished up the chapter 8, chapter transport, about the plant translocation. Okay, so for today we will enter chapter 9 lah. So maybe I will try to finish up the semester 2 syllabus in June, like that. Because I see most of your school now is already enter chapter 9, right? And some of the school even already enter chapter 10. Okay, so maybe your school teacher more faster than me. Okay, so this chapter 9 we all call as control and regulation. So actually if you refer to the MPM report, MPM Sukatan, the syllabus, actually this chapter is quite long compared to other chapter, although it only contains 2 subtopics, 9.1 nervous system and also 9.2 hormone. But 9.1 nervous system is very long topic. You can see the outcome syllabus, there are total I think is 10, more than 10 outcome that you need to learn, and then mostly outcome is that you never learned it before in SPM, just little bit that you already learned. Okay, so for today we will learn about the nervous system first, which is the first topic of the this chapter. Okay, so before we learn about the nervous system, neuron tissue, still remember, right? Like you already learned in Sem 1, nerve cell, which is the neuron, is the very important component in the nervous system, right? Because if you still remember that cells is the basic organ, basic life unit, and then if the cell is combined together, we will form neuron, and then if all the tissue combined together, we will form system, nervous system. So neuron tissue is very important. So although it is not in the syllabus, in semester 2 it is not in your syllabus, but you should still remember about the neuron tissue, the structure of neuron tissue too. Because if you don't know it, later I explain about the electrical impulse transmission, later you will confuse about that lah. What I am talking about. So before you learn about the nervous system, you should remember that you already learned in semester 1, which are neuron tissue 2. Okay, so this one is the basic structure, right? Basic diagram of the neuron cell. So if you still remember that there are 3 main classes of the neuron tissue, which is first one is multipolar, bipolar and unipolar. But normally that you mostly see is multipolar and also unipolar. Bipolar are very rare, and then that is not in our syllabus too, that you not learn in semester 1 too. But in semester 1 you already learn about multipolar and unipolar, right? Multipolar is the example is the motor neuron and also interneuron. So what are the difference between multipolar and dipolar? You see the cell body of the location. So normally polar neuron, the cell body is located at the end of the neuron cell. But for this unipolar, the cell body is located at the middle of the neuron. And then this unipolar is we call as sensory neuron. So you will see this lah. So normally in SPM that you already learned is about the neuron tissue, you learn about sensory neuron, right? Sensory neuron, interneuron, and also efferent neuron. Efferent neuron, these three types of cell. So normally sensory neuron is one type of the example of the unipolar. Okay, so what are the difference between sensory neuron and efferent neuron? You see the cell body location. So normally the sensory neuron, the cell body location is at the middle one, at the middle part. But for the efferent neuron, your cell body location is at the end, at the end of the neuron tissue. And then another difference is the length of the axon, and also length of the dendrite. So if you still remember that dendrite is the nerve that receive the electrical impulse. So for sensory neuron, all these from skin until the cell body is we call as dendritic cell. So this part, all this part we call as dendritic, meaning that this is the part that the electrical impulse is received from the skin toward the cell body. So once there afterward the cell body, it will become axon. So if you see the sensory neuron, the dendritic cell, dendritic length is more longer compared to motor neuron. So you will see the motor neuron, the dendritic cell is very short. Because you see that the cell body is already located at the end of the neuron cell, right? So dendritic is more shorter. And then once afterward the cell body, all of these are we call as axon, because axon is receive the electrical impulse from cell body and then transmit the electrical impulse to another type of neuron tissue through the synapse, or transmit the electrical impulse through the neuron, through the synapse to neuromuscular junction. So if through the synapse, this one, right, this one, so the location between the gap between the interneuron and also sensory neuron we call as synapse, right? This one you already learned in SPM. And then the location between the gap between the efferent neuron and also effector we known as neuromuscular junction. So these two structure, later we will learn more detail about the synapse and also neuromuscular junction. Okay, so this is the more detailed structure about the neuron tissue. So normally in neuron tissue you need to know about cell body lah, cell body, dendritic cell, and then also axon. So this is the example of the motor neuron, or we call as efferent neuron. Efferent neuron, that which is the at the end of the one end of the neuron cell is containing cell body, and another end of the neuron will be connected to the muscle tissue, and then the gap is we call as neuromuscular junction, that later you will learn it. Okay, so not only that, you will see that between the axon, you see that there are the cells we call as STPM cell. STPM cell, or another name you see we known as myelinated neuron tissue. So myelinated neuron tissue is, you see there are many STPM cell bounded to the axon of the neuron tissue. So if this is the very important structure in terms of the transmission of electrical impulse. Later we will learn it, but okay. And then between the gap between the STPM cell, we go known as the knot of Van Veur. Knot of Van Veur is the gap between the STPM cell. So this is very important. And then for this one is not very important. But the most important is this line lah, that you need to know it. And then this is all about the explanation about the neural tissue. I think I don't need to explain much. Okay. This one is I need to mention lah. So normally the neuron axon is surrounded by the glia, we call as SCAN1 cell. So this SCAN1 cell is very important to form the layer of myelin sheet, what we call as myelinated neuron tissue. Okay, so normally this one is very important. I already explained for this one is myelin sheath normally is poor conductor, meaning that the electrical will not pass through, will not enter. Meaning that the electrical impulse will not enter inside the myelinated sheath. The electrical impulse will only pass through the part that does not contain the SCAN1 cell. So meaning that the electrical impulse will be transmitted, jump from one node of inverse to another node of inverse. Meaning that when the dendrit already receive signal and they go to cell body, after they reach the axon, since there are the myelin sheath at here, the electrical impulse will directly jump to node of inverse, and then it will directly jump until it reach the terminal branch that already what we say as reach the neuromuscular junction lah. So this is very important feature, that it is one type of the characteristic of nerve impulse, that the nerve impulse is transmitted from one jump to the node of inverse to each other. Okay. So this is enable the fast conduction in vertebral axon. So the electrical impulse will be transmitted in very fast manner lah, compared to the unmyelinated cell. Okay. So and then I think that's all for the neuron tissue. Okay. And then for this another type of cell we call a glial cell. So normally glial cell actually is the cells that support the neuron tissue and play the role in insulating the axon of neuron and regulate the extracellular fluid surrounding the neuron. So you know that neuron actually is one type of tissue. So this tissue is also need to have the support. This tissue cannot move around. You need to know that in our body system, whatever kind of body system, you need to have a support. So nerve system, the supporting of the neuron tissue is we call glial cells. So you see this diagram. So actually there are many types of glial cell lah. So one type is we call an epidermal cell, astrocyte, oligodendrocyte, microglia, and also SCAN1 cells is also are also the one type of the glial cell that provide the support and also insulating of result. Okay, so this one is not very important. You don't need to know each type of the cell. The most important is you need to know there are the presenting of this kind of cell inside the nervous system lah. Okay, so this is the explanation again. I already explained it. So yeah, okay. Just now the first part is just about the recap that you already learned in Sem 1. So that part you should not take wasting your time lah, because you should already know it because Sem 1 only, right? I think just past few months only, so you should still remember, not totally forget, right? You should remember. Okay, now we go to the basic nervous system in the human. Okay, so what is the nervous system that we found in the human? Okay, so normally in human there are two main types of the nervous system that you need to know: central nervous system and also peripheral nervous system. There are two main types of nervous system. So if you see this diagram, so this is the diagram that you need, that you may see it in SPM. So normally central nervous system, you already mentioned central, so it will located at the central one. So our brain and also spinal cord and also the nerve cell that located in the eyes or in the eye located that connected with the brain are be known as the central nervous system. Okay, so what is the function of central nervous system? The is the very important one. Both of the systems are very important. We will not say that peripheral is not important. Both of them are very important, but their function is different. So normally central nervous system is serving as a central control. Actually, it is act as the integrating center that integrate all the response. Meaning that it is like a CPU, that control anything. When they already receive impulse, like you central nervous system, you already receive impulse, like the manager, I can say, like the manager. Okay, manager, you receive the task. You know that I need to do this task. Okay, so that your manager what to do, you need to integrate it. You need to divide the task to all the unit members, like the sponsor unit, activity unit, or HR, human resources, or marketing department, or sales department. All that unit, you need to distribute all the tasks that you already get. You get the task from the CEO, or CEO asks you to do this thing. Okay, you know that you cannot do all that thing, so you need to separate the task into the separate unit. So it is the integrate all the information received. They know that central nervous system know that when they receive a signal, what kind of response should the body make. Okay, so it will know it. Okay, when it receive the A signal, it will respond A response, so that it will send the signal to the PNS, peripheral nervous system. So you see here, it will send the signal to the peripheral nervous system to do the response. Either what kind of response So A response maybe when you are cooling, when you are very cold, what response you will take? You will wear jacket, right? You will wear jacket. So not only the behavioral response, but your internal organ response will also take place. So normally peripheral nervous system contain two types of nervous system, which are the somatic division and also autonomic division. So normally somatic division is the external organ, but for autonomic division is normally internal organ. Okay. So normally this is the signal pathway. So normally you should have the sensory receptor. Every organ, not only skin, every organ should have the sensory receptor. So when they are the sensory receptor already stimulated, it will send the impulse through the sensory neuron to CNS, central nervous system. And then inside the central nervous system, interneural central nervous system, it will integrate all the information received. What kind of response need to do? What kind of response? Maybe contraction, relaxation, or secret. What kind of hormone? All this is CNS take place. Okay, so once they already integrate information, they already know what kind of response, it will send this kind of response stimulus through the effector neuron or motor neuron to the muscle. Not only muscle, maybe go to the gland or maybe go to the other parts, but normally it's muscle. So it will go to the muscle and then it will cause the response. Either is muscle contraction or muscle relaxation, any kind of response. But for example is one of the example is muscle contraction. Okay, so this is the very general process. So normally you see this diagram again. So I already mentioned, you will set when your internal stimuli is stimulated, sensory receptor will be stimulated, and then will send the impulse to the CNS through the afferent neuron, and then this central nervous system will be stimulated and integrate all the response received from the afferent neuron, and then transmit this integrated impulse to the efferent neuron. So afferent neuron and efferent neuron normally is located in the under the peripheral nervous system. So the central nervous system is very simple. The function is very simple only. It just receives the impulse from the afferent neuron and then integrate it, what kind of response that he need to make, the human need to make, what kind of hormone that the gland need to secrete, and what kind of the internal organ response need to make. So after that, they already integrate finish, it will send the impulse, this integrated electrical impulse response to all the nervous system, the peripheral nervous system back through the efferent neuron. So that you will see that when the efferent neuron, can be connected to the autonomic nervous system or somatic system. So normally somatic system, another name is we call as motor system. So normally motor system will be more focused on skeletal muscle lah. Yeah, it will more focus on skeletal muscle. But for the autonomic nervous system, it will either go to sympathetic or parasympathetic. So it will either go to sympathetic division or parasympathetic division. But at the end of the result, normally it will have one type of response. Either it is muscle contraction or muscle relaxation, but for this sympathetic and parasympathetic, normally it will involve of the gland or the internal organ stimulation. Your eyes, your pancreas, your adrenal gland, or liver, what kind of response should liver do, all that. So this is the very important that you need to know it. So this is the explanation. Okay. Next, I go to sympathetic and parasympathetic nervous system. So directly lah, because if you go to the syllabus, actually this topic is very at the end. But it's still even in syllabus. So I take in front of my class, so more easy to you lah, because I already explained about the peripheral nervous system and also mentioned about sympathetic and parasympathetic. So I go to the sympathetic and parasympathetic nervous system. Okay, so normally parasympathetic and sympathetic nervous system are the classes under the autonomic nervous system, which are also including under the peripheral nervous system too. Okay, so it is an autonomic nervous system. Autonomic meaning that it is automatically. Every time this this nerve cell either sympathetic or parasympathetic will be stimulated. Either one. Not say that both of them is totally rest. No, either one will be stimulated. Okay, so normally if you see this. Sympathetic and parasympathetic. So this is the general flow of the nervous system. Okay, so I already mentioned about that. In our nervous system, you know that we have afferent neuron and also efferent neuron, right? All this that you already know. So for this kind of cell, normally it will involve two efferent neuron. Both of them are efferent neuron, but the difference is we call as this one, that the initial part of the nervous system is we call as preganglionic neuron, and then at the center gap, the interaction between the neuron we call as ganglia. This part the terminal branch and also the cell body part we call as ganglia, and then after ganglia we known as postganglionic neuron. Okay, so the main difference you can see this diagram is preganglionic and postganglionic structure are different. Preganglionic is myelinated structure, but postganglionic is unmyelinated. You see that totally axon only. It only contain axon. Does not containing the myelin sheath, but preganglionic have. So this is the difference between preganglionic and postganglionic. And then what are the difference between the sympathetic and parasympathetic nervous system in terms of structure? You see that preganglionic for sympathetic is more shorter. more shorter compared to parasympathetic, because parasympathetic more longer, right? More longer, you see here, longer. But so for sympathetic, pre-ganglionic is shorter, but post-ganglionic is longer. For parasympathetic, pre-ganglionic is longer, but post-ganglionic is shorter. This is one type of difference in terms of neuron structure. And then another difference is in terms of the neurotransmitter they release. In both of the systems, sympathetic and parasympathetic, at the first part, for the pre-ganglionic part, the interaction between the pre-ganglionic and post-ganglionic, they will release they release the same neurotransmitter we call as acetylcholine. Both of them release same. But at the end of the system, post-ganglionic neuron, the neurotransmitter released are different. For the sympathetic, the neurotransmitter release is norepinephrine, or another name we call as noradrenaline. Noradrenaline is British style and norepinephrine is American style. So, but both of them are acceptable in exam. And then parasympathetic will release acetylcholine. So this is another second difference. Very important. The first difference is neurotransmitter. Important is the length between the pre-ganglionic and post-ganglionic neuron. Second difference is the neurotransmitter that release at the final terminal, at the post-ganglionic neuron. Not pre-ganglionic neuron, both of them release same neurotransmitter, which are acetylcholine. But at post-ganglionic neuron that already attached to the effector, the neurotransmitter are totally different. Post is for sympathetic is noradrenaline. For the parasympathetic is acetylcholine. Okay, so this is very important. And why? Because this one is one of the topic that you mostly student will not focus. Yeah, why? Never come out in SP exam before. In structure question and also in essay question, never come out. But in objective question, yeah, come out lah. But in structure and essay question never come up yet. So I hope that you should not avoid it lah, because it's very easy to understand only. Okay, and then this one is very famous about the response. What kind of the difference between the response between the parasympathetic and sympathetic? So normally, if you still remember that you already learned in chapter 8, regulation of heartbeat, as I teach before. If you still remember that, about the regulation of the heartbeat rate, one of the factor that regulate the heartbeat is nervous system, right? So you see here, for parasympathetic, you know that when the nervous system release the neurotransmitter we call acetylcholine, it will slow down the heart, right? In parasympathetic, if you release the acetylcholine, it will slow down the heartbeat. But in sympathetic, it will increase the heartbeat. Why? Because it release noradrenaline. Noradrenaline, right? And then the mechanism I also explained in chapter 8 too. But later I will explain one more time at the following part. Okay, so the one type of the very famous question, also inside between the difference between the parasympathetic and sympathetic. So normally in parasympathetic, very easy. Parasympathetic is in rest and digest condition. At the rest, like now, rest, heart, or digestion condition, parasympathetic division will take place. But in sympathetic, you know that sympathetic, what it will release as noradrenaline. So noradrenaline will increase your stress response, right? you will make you more stressful, more panic, and more concentrate. Why? Because sympathetic is responding to dangerous and stress condition. Why? Because as fight and flight response. When you are very stressed, when you are in danger part condition, then sympathetic will be take place. So these are all the response through the organ that we found in the human. So normally in sympathetic, you need to dilate your eye and then inhibit your salivary gland. Yeah, you know that when you are very panic, very stressed, very dangerous condition, or very nervous, but you will not have a very your salivary is very low. Why? Because it already inhibit your salivary gland and also relax your bronchus in your lung. You need to relax because you are very stressed. So you need to relax. Starting to relax, eat, and then accelerate your heartbeat, inhibit your activity of stomach and intestine. So normally, yeah, if you see that the people in very concentrated concentrated people like doctor, you will see the doctor in surgery. The surgery doctor, they can stay at the surgery room or operation room and make an operation until 10 hours or even 12 hours, 18 hours without drinking or eating. Why? Because it inhibit your activity of your stomach. Or as you student, if you are very concentrate to study, concentrate to your study, you also not have the what you say takkan rasa kenyang lah. Awak takkan rasa kenyang punya. Once you relax, then yeah, you akan rasa kenyang. Rasa kenyang. Kenyang. You will face hungry. You will become more hungry. So why? Because of the sympathetic division. And another one is inhibit your pancreas activity, and then also starting to release glucose. So that is why that the people in very stress condition, you will more easily to increase your weight. Although you are not eating or you are not eating much, but it will increase your weight too, because your blood glucose level is very high, because it need to stimulate your glucose for increase your metabolic activity, because you are now very stressed too. So you need to have the high amount of the glucose used by the cell to maintain the high metabolic activity. And also stimulate adrenal medulla, inhibit emptying of bladder, and also promote the education and vaginal contraction. Yeah, and then for parasympathetic is a reversible action from the sympathetic. Okay, so that's all for the differences between parasympathetic and sympathetic. Okay, so any questions so far? So I think no question. Okay. Okay, now I will proceed to the formation of resting potential. Okay, so what is it mean? So it is meaning that the neuron is resting state that does not sending any electrical signal to you guys, within the neuron, meaning that neuron is at resting state. No electrical impulse will be passed through. So if you still remember about the structure of neuron, you still remember the structure of neuron. Electrical impulse will be transmitted from one place to one end, from the beginning to the end, and then you can imagine that the neuron tissue is like the wire. You know that wire, right? Why we want to wire? Because we need to have electricity, right? So it like a wire between the inside of our body. So normally the electrical impulse will be transmitted from one place to another place, right? So when the electrical impulse is transmitted, this is what we call as action potential. But the electrical impulse will not always be occur inside the neuron one. So when there is no electrical impulse inside the neuron or inside within the axon, then this one we call as resting potential. But when there are the electrical impulse within the one part of the axon, you need to know that electrical impulse is not directly have throughout the axon. The electrical impulse is transmitted from one part to another part. So when this part have the electrical impulse, this part will be rest, and when this part have electrical impulse, this part will be rest. So normally when the rest is we call as resting potential, and then got the electrical impulse we call as action potential. Okay, so normally in resting potential, if you have about the electrical impulse, then voltage is the key component. Voltage, or we call as potential difference, that you already learned in physics. So although you are not taking physics or physics, maybe, but in SPM I think you already learned about the definition of the voltage, right? The definition of potential difference. So normally, if you still remember that when they are the electrical charge between between the two region, then potential difference or voltage will be formed. So normally membrane potential actually is voltage. Membrane voltage actually is the differences in the concentration between the specific ion inside the cell and those outside the cell. So normally is in the structure of the axon, if you still remember, in the structure of the axon, within the axon is inside the cell, but outside the cell, this white color region is we call as outside the cell, or we call as extracellular fluid region. So this is outside the cell. So the outside the cell and inside the cell, the ion, the charged ion is different. So normally sodium ion and potassium ion are the key ion that play the role in the membrane potential. So these two ions are always maintain the concentration gradient during the resting state. So if you still remember, sodium and potassium pump, right, at they transport what they transport. So the sodium and potassium pump is regulate your sodium ion and also potassium ion between the cell during the resting state. So if you still remember that in sodium potassium pump, you know that sodium ion inside the cell will be pumped out outside the cell, right? 3 sodium ion pump out outside the cell, and then 2 potassium ion pump into the cell, and then this process is require ATP, one type of ATP transport. So why? So if you see that, meaning that outside the cell, sodium ion is high concentration, but inside the cell, potassium ion is high concentration. Okay, so you see this potassium concentration normally inside the cell is high, but sodium ion is high outside the cell. This normal situation. At equilibrium situation, sodium ion at outside is high, and inside the cell, potassium ion is high. So this asymmetric distribution of ion across the plasma membrane in the vase is both about the action of selective ion channel and also ion pump. Okay, so not only sodium potassium pump, actually there are also the what we call as ion channel that specifically for potassium ion and also sodium ion. So you see this diagram, this blue color is sodium potassium pump, and then this more orange color, more deep brown color is we call as potassium ion, and then this yellow color is sodium channel ion. Sodium ion channel. So you see this diagram, you see that the number of potassium ion channel is high quantity compared to sodium ion channel. Why? Because you need to maintain that all the outside the cells, sodium ion is high. Inside the cell, potassium ion is high. So Normally this is the ion channel, so this is not anti-transport process, this is the facilitated diffusion process. So the ion will be moved down the concentration gradient. So normally the sodium ion inside the cell will be passed through out sodium ion outside the cell will be passed into inside the cell, because down the concentration gradient, right? Because it need to maintain the ion, and then the potassium ion inside the cell will be passed through out from the cell. So that is why that your potassium ion channel need to in high amount, because you know that potassium and sodium ion both out there are positively charged, and then in resting potential, your inside the cell, inside the axon, within the axon should be negatively charged. Resting potential must be negatively charged. Outside the cell is positively charged, so that the potassium ion need to pass through out. Kena keluar punya. Dia kena keluar. Tak boleh terlalu banyak juga. Kalau terlalu banyak pun akan possibility to create the positively charge too. So dia kena keluar. And then the sodium ion tak boleh banyak. The sodium ion channel cannot too much. If too much of sodium ion channel, so sodium ion will be flushed into the cell. So yeah, it will become positively charged. Cannot. So normally ion can be filled rapidly through ion channel, causing a net movement or charge generate membrane potential. So normally ion channel will convert chemical energy to electrical energy due to they are selectively permeable. Okay. And then potassium ion channel are always open, causing the potassium ion diffuse out from the cell. And then you already mentioned, yeah, in the potassium ion inside the cell is very high. I already mentioned inside a cell is very high, so that the potassium ion will be outside the cell. Okay, and then only a few sodium ion, and then ion. Okay, and then another thing is that there are also another type of channel we call as voltage gated ion channel. So this is also one type of the ion channel, but this kind of channel can be open and closed or closed. This one is opening channel, totally opening 24 hours. They cannot close one. So meaning that the ion can be freely move through this channel based on the concentration gradient. But for this one, voltage gated ion channel, the ion cannot pass through it when the channel is closed. Only when the ion channel is open, then the ion can pass through. So when the ion channel is open, when your membrane potential is changing, meaning that when the charge are changed very drastically, meaning that there are significant change between the charge between the outside the cell and inside the cell. When there are the significant change, then this gated channel will be open. And then in the resting state, we will known as this state, the axon is polarized. The axon membrane is polarized because your inside the cell is negatively charged. The most important thing is resting potential inside the cell is negatively charged because no electrical atmosphere. Only outside the cell is positively charged. Okay, so this is all about the resting potential. Okay, so when we finish up the resting potential, then we will need focus on action potential. So action potential is where when there are the electrical impulse. So just now I say that resting potential does not have the electrical impulse, right? So if there are no electrical impulse, that is resting state, so in normal condition. But for now, when there are the electrical impulse pass through the axon, action potential will be triggered. So it is the electrical signal that the travel rapidly down the axon into the synaptic terminal, which is synapse lah. Okay. So this is the overall process. So there are actually total 5 processes lah. So this is one type of gut and another type of gut at here. So first step is resting state lah. So I already mentioned it. When there are resting state, there are totally no membrane. There are totally no membrane potential. The membrane potential must be negatively charged. I already mentioned that in resting potential, your axon membrane is negative. So if totally negative, you see that it is totally negative one. But when the action potential is starting to be triggered, when when there are the electrical signals starting pass through the axon, then depolarization will be starting to occur. Depolarization. Okay, so meaning at the depolarization. Okay, so when after the depolarization, when the voltage is reach the threshold level, it will have the reach the threshold level, it will increase. Your axon membrane voltage now will be starting to increase. When the axon membrane potential starting to increase and reach the threshold level, it will directly trigger the action potential rising state directly. When it reaches the threshold level, action potential directly be triggered out. Okay, once the action potential is triggered, then it will flow back. So it will be called as repolarization. Repolarization, meaning it was starting to polarize back again to form to the resting state. So meaning that the membrane potential was starting to decrease until negative, and then you see that it will decrease more negative compared to resting potential. Because you see that resting potential is at the maybe is negative 70, but during the repolarization state, it will decrease down lesser than more negative than negative 70, maybe negative 80, negative 90 like that. So this process is we call as undershoot, more negatively compared to resting potential. Okay, after undershoot, it will back to the resting potential again. So this is the overall process of the action potential. So we will look it one by one. So resting state, I think I don't need to explain much. Right. So you still remember we have 3 types of channel, right? 4 types of channel actually. One type of channel is sodium potassium pump, right? Sodium potassium pump. Second type is ion channel, sodium ion channel and also potassium ion channel. But these 2 channels are totally open. And another type of channel is sodium is the voltage gated ion channel. So voltage gated ion channel is that this channel can be closed and open. So there are also 2 types of sodium and potassium. So for sodium is only allow sodium ion to pass through. Potassium ion is only allow potassium ion to pass through. So in resting state, all the voltage gated channel are closed. All of them are closed. So meaning that the sodium ion cannot pass through and your potassium ion also cannot pass through. Okay. But when there are the nerve impulse, when the nerve impulse is starting to arrive in one part at the beginning of the axon, then your sodium gated ion channel will start to open. But not all, few number only. Only few number only will open. Not all they will be open, some of them. So that sodium ion will be passed through into the axon. will be influenced through the channel, causing depolarization. Meaning that why causing depolarization? Meaning that your sodium ion channel is highly positively charged, right? So when they enter inside the cell, meaning that your inside the cell will start membrane potential starting to increase already. Start to increase because there are many sodium ion channel is already starting to enter into the cell already. So if but it is based on the stimulus too. So if the stimulus or nerve impulse is very strong, so directly more gated ion channel will be open. But if very weak or normal one, only few number of sodium ion gated channel will be open only. Okay, so this is what we call as depolarization, meaning that the membrane potential start to increase and the sodium voltage gated ion channel sodium voltage gated ion channel start to open. Okay, once the membrane potential is already reached the threshold level, once it reach the threshold level, it will directly go to rising state, meaning that action potential will be start to trigger. Once it reaches the threshold level, then rising state directly start. So in the rising state, meaning that at this point, your axon membrane potential will be increased very high, increased very linearly, meaning that your inside the cell will be positively charged. So also why is positively charged? Because all of your sodium voltage gated ion channel all are open. All of them are open. So more sodium ion, all the sodium ion outside the cell will influx into the axon. So when they are influx into the axon, so that inside the cell containing many sodium ion, so that your cell will be positively charged. But at the same time, your potassium gated ion channel will remain closed. Dia tak akan buka. Okay, so once they already reach the action potential, then it need go back to resting potential again, because electrical impulse it already passed through it. You need to know that this one is already electrical peak. Once the electrical peak impulse is reach the certain part of the axon, so your action potential is triggered. But when the electrical impulse is already passed through, already go through it, then this part of the action will not have the electrical impulse. Then the membrane potential inside the axon was starting to decrease. So meaning that your sodium ion gated channel is inactivated and starting to close. And then at the same time, your potassium ion gated channel will be open, meaning that your sodium ion cannot pass through it, into it, and then your potassium ion channel will be open, meaning that all the potassium ion inside the cell will be efflux out from the cell. So this one will cause that your charge inside the cell was starting to become negative. Yeah, right, because potassium ion also is the positively charged ion. So all the potassium ion is already is coming out from the cell. Then although it containing sodium ion, but since the potassium ion is already come out, so that the ion inside the cell is also starting to become negatively charged. Okay, so once the membrane potential inside the axon starting to decrease until it reach the resting potential state, so maybe it reach the here, then it will decrease again. This what we call as undershoot or hyperpolarization. So what does it mean? So meaning that the membrane potential permeability to potassium ion is very high, meaning that your membrane is very high permeable to potassium ion. For the repolarization, it's just based on the normal concentration gradient. So just based on normal concentration gradient, because you know that potassium ion is already accumulated at here, so all the potassium ion will be flushed out, and then to achieve the equilibrium only. But for undershoot process, it will make your membrane, axon membrane, become highly permeable to potassium ion, so that your potassium ion will be influx out from the cell to maintain the negatively charged inside the cell. Okay, so that it need to make sure that inside the cell is totally negatively charged. So this reaction is recalled as hyperpolarization process. Okay, so once all the potassium they maintain that, they realize that the axon membrane now is totally negatively charged, so that undershoot process is end, the membrane potential will increase back again to the action to the resting state, so that the resting state, and once they reach the resting state, your potassium ion channel will close again, and then this process is repeat and repeat again. Dia akan repeat dia akan maybe repeat again, and then repeat again, because it is transmission along, right? It will transmitted along the nerve impulse through the axon. Okay, so this is the theory about the action potential lah. Okay. Okay, next I will explain about the characteristic of nerve impulse. Okay, so I already explained about the nerve impulse that it can be transmitted along the axon, and then when the one part of the axon receive the electrical impulse, it will trigger the action potential. Okay, now we need to learn about the characteristic of nerve impulse. Okay, so what is the characteristic of nerve impulse? So first one is refractory period. Nerve impulse is transmitted through the refractory period, meaning that what is the refractory period? When the sodium ion channel remain inactivated during the falling phase and the early part of the undershoot, meaning that this one. Refractory period is the period your sodium channel is inactivated during the falling phase and early part of the undershoot. So at here, this is what we call as refractory period actually. Okay, so the second depolarizing stimulus occur during this period, it will unable to trigger the action potential. So meaning that this period actually is to prevent frequent action potential, meaning that when there are the action potential, when the membrane potential is starting to decrease, at the same time, when they're starting to decrease, at the same time there are another type of stimulus again. So when there are another type of stimulus again, this stimulus will be inhibited, akan stop dia. Why? Because of refractory period. Kerana dia kena siapkan semua ni, it want to finish one round, then it can trigger another type of action potential. If not, during the rising state of repolarization or in the undershoot state, if there are another type of stimulus occur at the same time, then no action potential will be triggered again. Dia tak akan keluar sini akan naik atas, naik balik, tak ada. This is one of the refractory period function. So the downtime when a second action potential cannot be initiated is called the refractory period. So this period is the safety zone to prevent that the stimulus is too frequently triggered. If there are too many, if you will be imagine that if during repolarization start to decrease and then got stimulus again, meaning that the membrane potential will start to increase again and trigger another type of potential higher than this. Cannot. No. So you need to inhibit this. So refractory period is due to inactivation of sodium ion channel, not the charge in the ion concentration gradient. It is very important. You need to know that refractory period is due to your sodium ion channel is starting inactivate rather than the ion concentration gradient. Not say that when potassium ion is influx out, then this we call as refractory period, or sodium ion inside out, or the sodium ion is blocked, or the charge between the axon and the outside the cell is different. It we call as refractory period. No. And then the flow or charge particle deviation potential involve far too few ions to change the concentration either side of the permeable membrane significantly, and then it only allow the action potential move in one direction. This preferably be only one direction. When there are the stimulus at here, dia tak akan terbalik punya. Cannot, you will not go to the reverse direction. But if go to reverse direction, then got problem lah. Okay. And then it also limit the frequency of action potential when success free. This is what I just mentioned about it. Limit. We cannot too high. If too high, you imagine that it is the voltage. You know that voltage is too high. If you see that like the too high voltage, it will make the people die, right? Like the electrical shock from the blood. From the blood, you may die, right? If the why? Because the voltage is too high. When the voltage is too very high, it can lead to the vital case, meaning that you will die. So it need to inhibit. So you see that the membrane potential cannot too high one. Maximum is only 30. Cannot too high. If too high like 50, 60 or even 100, wow, you will die. You will die. You will face the electrical shock. So this action potential is limited. Dia nak limit. Kalau tak limit, dia datang, then your membrane potential will be very high until can reach 100, 120, even though until 240 if you not limited. So this is very important. Okay, and then there are two types of refractory period. It is solely refractory period and relative refractory period. So you will see this diagram more easy lah. As solely refractory period is between the rising state and also depolarization and also repolarization, and relative refractory period is in the undershoot, or we call as hyperpolarization state. Okay. Another characteristic is all or none law. Okay. What are these meaning? Okay. So normally, if you see this diagram more easily to me explain, meaning that all of none law, either it can be triggered, the action potential either can be triggered or cannot be triggered only. It does not containing that only half type of action potential. No such thing. Either it can trigger, no matter your electrical impulse, your stimulus is too high. When your stimulus it can make the membrane potential reach the threshold level, then it will directly trigger action potential. So maybe your stimulus is although it is very weak, if it is very weak, meaning that it does not reach the threshold level, then no further depolarization process, repolarization process. Okay, so it will directly go to the resting state lah. So no action potential will be occur. So this is the all or none law. So this is the explanation you can read yourself. Okay. And then about the speeding. Okay, about the speed, normally that the control the speed is one is about the diameter and also one is about the presence of myelin shift. So normally in diameter of axon is when the diameter of the axon is more larger, then you will have a faster speed of conduction lah. So why? Because because the smaller diameter will have the greater resistance, and it will lower the speed. Because if you imagine that when the diameter is long, then it will have more space and less resistance. And that also present on myelin sheet. So this one I already explained before lah. Why? Because it containing myelin sheet made up of scaffold cell, because it's a good insulator, so no electrical will be passed through. So normally the electrical will be propagated, will be moved through the node of Benville only. So the action potential of a myelinated axon will be propagate more rapidly, because time consuming opening and closing are very short, and then this mechanism we call as secondary conduction, because the action potential is appear jump from node to node along the axon. Yeah. Okay, so that's all for about the transmission characteristic of the nerve impulse. Next, I will explain about the transmission along the transmission of the action potential or transmission of the nerve impulse along the axon. Okay. So I already explained about the action potential. So everyone should know what is the meaning of action potential and what are the mechanism of the action potential. Now I need to explain about the mechanism that the action potential is transmitted along the axon. So as I mentioned before, the action potential, the electrical charge, the nerve impulse will not directly pass through directly one. It will pass through one part to one part. So meaning that when the nerve impulse reach this region, only this region will trigger action potential only. Other region still maintain negatively charged. Okay, so you see there, when the nerve impulse is trigger action potential, so this is the action potential lah, so that the sodium ion is influx into the axon. Okay, so now this region, A region, I say that the A region, it will become action potential. Okay, once that this action potential actually depolarization of action potential. So you still remember that after the action potential, depolarization process will be take place. So when the depolarization is take place, at the same time it will spread the action potential to the neighboring region. Meaning that depolarization of A region will cause the B region to trigger the action potential and reinitiate the action potential there, and then the A region will start to depolarize again and return back to resting state. And then to left of this region, the membrane is repolarizing as potassium ion as outflow. And then after that, this depolarization and repolarization process is repeated. So meaning that at here is depolarization process, right? Okay, so once it already had action potential, it will enter repolarization process. Okay, so when the enter repolarization process, it will initiate again C region. So like this, it will initiate again the C region to trigger the action potential inside the C region, and then the B region will be at the repolarization process. So this is the repeated process. So depolarization will trigger the neighboring region to initiate the action potential of the neighboring region. Yeah, so this is the concept. So this is the explanation actually. So when the action is initiated, so it will create the electrical current, or we call as localized current, right, to depolarize the neighboring region. Yeah. So regions. Yeah. Okay, so when the sodium ion channel is already at here, so actually it will trigger the electrical impulse and starting to depolarize this region. Okay, Okay, so normally at the beginning location, okay, and then the localized electron will cause the depolarization of the beginning location and split this localized electron will be split to neighboring part of the membrane, meaning that neighboring part of the membrane action potential is initiated by depolarization of membrane. Depolarization of the membrane. Yeah, and then depolarization of membrane. So after the action potential, membrane is repolarized back to resting state, right? So this depolarization and repolarization potential is repeated. So I repeat again, when the action potential is triggered, depolarization depolarization is triggered, right? And then this trigger depolarization process will trigger the localized electron. So it will starting to initiate B region to trigger the action potential already. Initiate, but only, but not trigger yet. It was starting to trigger when the A region back to the starting the V polarization replay RE. When it go to the V polarization state in the A region, then B region will have the action potential. We're starting to depolarize, and then B region depolarize, it will also create the localized electron initiate C region to trigger action potential. And then once the B region go to V polarization V polarization, it will trigger the action potential of the C region. So this depolarization and repolarization process are repeated again and again until it reach the synaptic cliff. So after the action so this means that the local cover of ion, it cause the plasma membrane, cause the action potential. So there are the localized electron at there. So localized covalent only polarize the axon membrane ahead of action potential, but not produce another action potential, meaning that actually it is to reinitiate the action potential again, but not produce another type of action potential, just continue only. Meaning that at the B region, it just continue the similar type of action potential from A only, meaning that the action potential, the membrane potential for this B region is similar to A1. If A is 30, then B is also 30. If A is 20, then B is also 20. Similar one, no change one. So you cannot say that B region is trigger create new potential. If you say new action potential, then totally no marks as well, because it repeat only. So you need to remember that. Okay, so I think maybe that's all for today. So I already explained about the neuron. So maybe I will proceed to synapse. This is the last topic today. So any questions so far? Okay, so no question, so I will proceed it. Okay, so normally synapse, I think everyone is already know, right? What is the synapse that you already learn in SPM 2? Okay. Anyway, okay, if you already forget, never mind. You make a revision again. Okay, so normally synapse is the junction or the gap between the two neurons, like the sensory neuron, between the sensory neuron and interneuron, and interneuron between and the motor neuron. So all these is a gap be known as synapse. Okay, so you see that this is one A neuron, and then this is opposite neuron, B neuron. So this is what we call as presynaptic axon and postsynaptic cell. Presynaptic neuron and also postsynaptic neuron. So presynaptic is the electrical impulse coming in, and then postsynaptic is receive the electrical impulse from the presynaptic axon 1. Okay, so normally you need to know the structure. So neuron that terminate synapse is presynaptic, and then terminate is presynaptic, and then begin at synapse is postsynaptic, and then shrilling end is known as synaptic knob or synaptic cleft, is both are acceptable, and then separator or synaptic knob containing synaptic knob is at this one is we call as synaptic knob, sorry, and then the gap between the neuron is we call as synaptic cleft, and then cytoplasm containing mitochondria lah, where we know that because it require energy, and then we know that it containing neurotransmitter, right? Neurotransmitter, because cytoplasm containing neurotransmitter. So normally in the presynaptic neuron synaptic membrane, synaptic knob, the neurotransmitter is enclosed in the synaptic vesicle. Okay, so this is just a general structure about the synaptic structure synapse lah. So this one I think I don't need to wasting the time at here. So impulse transmission, I think this one is you are also learn in STPM 2. So normally if you talk about the impulse transmission, so normally when the electrical impulse is already reached the synaptic knob, when it already reached the synaptic knob, so that it will trigger the synaptic vesicle to release the neurotransmitter. But before that, you know that depolarization and repolarization process, right? So action potential still will be occur. So when the action potential is occur, done, so depolarization will cause the calcium ion channel inside on the presynaptic membrane open, and then calcium ion will be influx move into the presynaptic synapse cytoplasm. Yeah. So when the calcium ion is already inside the synaptic knob, so that the calcium ion will cause the synaptic vesicle fuse to the presynaptic membrane and starting to release the neurotransmitter. So any type of neurotransmitter, so just now I already mentioned, right? One of the example is acetylcholine and another one is noradrenaline, right? Okay. So this neurotransmitter later it will bind toward the receptor that located on the postsynaptic membrane. Yeah, so you see this diagram. So it will bind to the receptor at the postsynaptic membrane, and then it will trigger the action. So not necessarily about the sodium potassium pump, not necessarily it can be have another type of reaction. But one of the example is trigger sodium potassium pump lah, meaning that sodium ion is pumped into the cell and then potassium ion is pumped out from the cell. Yeah, to trigger the action potential of the postsynaptic membrane. Why? Because postsynaptic membrane. Okay. Okay, so the another one, yeah. Okay. And then this is the explanation, so I don't need to explain much. Okay. Okay, so I need to explain about the activated receptor with same impulse to postsynaptic neuron. Okay, so when the neurotransmitter is already bind to the receptor on the postsynaptic membrane, it will trigger the action potential of the postsynaptic neuron. So this receptor will send the impulse to the postsynaptic neuron to trigger the action potential, and then the impulse that we will have is two types of impulse that we need to learn is EPSP and also IPSP. EPSP is excitatory postsynaptic potential, and also IPSP inhibitory postsynaptic potential. So excitatory EPSP, you will see the excite. So meaning that it will change the membrane potential change, will bring the closer to firing, meaning that this one will trigger the action potential one of the postsynaptic neuron. But for this one, it will inhibit the action potential for the postsynaptic neuron. Meaning, you see that action take the neuron farther away from the firing level. So firing level actually is we call it action potential lah. Okay. Okay. So in EPSP, so normally the neurotransmitter role is acetylcholine. So normally it will bind to the receptor and permeable to both potassium and sodium, and then the sodium ion will be influx into the membrane to depolarize it so that it will trigger the action potential. But IPSP is different. It would inhibit the action potential, meaning that you see this. One of the example of the neurotransmitter is GABA. Later we will see it, the full name GABA. So this neurotransmitter is reversible compared to acetylcholine, because acetylcholine is trigger action potential, but GABA is inhibit action potential, meaning that it will more permeable to the ion we call as potassium ion and also chloride ion. Alright, ion. Okay, when this ion channel are open, the postsynaptic membrane is hyperpolarized. Oh, you still remember about the hyperpolarized, meaning that it will more negative compared to resting state. So it will totally inhibit the action potential of the postsynaptic neuron. So it will move the membrane potential further from the threshold level, and then inside the cell is negatively charged, so no action potential will be triggered. This is what we call as IPSP. Okay. And then after the neurotransmitter is released out from the presynaptic neuron, then the neurotransmitter cannot always maintaining at the in the synaptic cleft. The neurotransmitter need to recycle back. Dia kena hilang punya. Dah tak boleh selalu dekat. Cannot always maintain at the synaptic cleft one. It need to recycle back or disappear one. If the neurotransmitter is still maintaining at here, meaning it will trigger the action again and again. Yeah, because neurotransmitter is still remain at here, so it will bind again. You need to know that when the neurotransmitter bind to the receptor on the postsynaptic neuron, once the action potential is triggered, this neurotransmitter will be loose by link. Dia akan keluar, but dia kena disappear daripada synaptic cleft ni. Hilang daripada synaptic cleft. If not, it will trigger again and again. So you need to terminate the neurotransmitter signaling. So there are two ways to terminate it. Either using enzyme, you use the enzyme to break down it, or the neurotransmitter automatically flow back into the presynaptic neuron through the transport channel. Two ways, either using enzyme or using the neurotransmitter transport channel, recycle back to the presynaptic neuron. Two ways lah. Okay. And then about the summation of postsynaptic potential. So this is about the EPSP and IPSP that learned before and learned just now. Okay, so normally in synapse in the postsynaptic neuron, if you want to trigger the action potential, you still need to make the action membrane potential reach the threshold level. Then you can trigger the action potential in the postsynaptic membrane. But normally in the synapse, due to the synapse got the gap right, so normally the impulse at the postsynaptic membrane is quite low compared to presynaptic neuron, because it already got the gap there. So the electrical impulse normally is very low. So even though when your electrical impulse is EPSP2, like this EPSP, but if your electrical impulse is very low, that not reach the threshold level, then postsynaptic membrane also cannot trigger the action potential. Also cannot trigger the action potential. Only when your membrane action potential reach the threshold level, then it will trigger the action potential on the postsynaptic neuron. So how to trigger it? You will see this first situation, no summation, meaning that the impulse from E1 and E2, you see E1 and E2 is different, is only from E1 only, meaning that the nerve impulse is only coming from first part only, this part only. Other type of dendrit does not receive any impulse, only E1 receive impulse, and then the impulse received is separate time. The first impulse is maybe at 1 second, and another 5 seconds E1 still receive the another E1 impulse, but these two impulse is too far away. The period for this both impulse are too far. Like maybe the first E1 come, but another E1 need to wait about 30 seconds. So when you too long, the duration, the gap is too long, so it cannot reach the threshold level, so it cannot trigger action potential for postsynaptic neuron. You need to know this action potential is triggered for postsynaptic neuron. Okay, but if they are the temporal summation, meaning that when there are two types of the nerve impulse coming from the same synapse, same synapse, and then the impulse, the gap between these two impulse are very short, so maybe 2 seconds, 1 second, or 0.5 seconds, very fast, so that it can combine it together and it will reach the threshold level and trigger action potential. So this one is we call as temporal summation. This one will only involve one type of synapse only, one synapse only. But there are also the spatial summation, meaning it involves two synapses. So maybe one synapse at here and one synapse at here, and they combine together at the same time. You need to measure at the same time. If two get like this, then no also lah, no action potential. But if at the same time too, we know as a spatial summation, so that it will reach the threshold level and trigger the action potential of the postsynaptic neuron. Final one is partial summation, but it will involve IPSP. So when it involves IPSP, you know that IPSP is inhibit the action potential. So once it be involved, although you have EPSP here, but since got IPSP at another part, so it will inhibit the trigger coming out. Coming out is depolarization, right? Depolarization, right? Why? Because why? Because in depolarization process, sodium ions coming in, right? And coming in, right? And then you need to maintaining the high positively charge inside the cell. So potassium ion channel, another way is potassium ion channel can be open and coming out, so that it can increase the rate of depolarization and increase the permeability of potassium ion to the membrane. And another one is noradrenaline. So normally this one function is to involve in parasympathetic nerve effect. So normally this one is involve the CMP activation, and then it will get the calcium ion to stimulate it, and then it will trigger the depolarization process. And another one is dopamine. So dopamine actually is not in our syllabus, but later you will learn about the cocaine mechanism. So you should know too lah. So it is one type of neurotransmitter that normally is play the role in excitation and inhibition process. So normally this one or neurotransmitter can be inhibited by cocaine. One type of drug, right? Cocaine, I think everyone is familiar with it. What is cocaine? So later we will learn about the cocaine drug mechanism inside in our nervous system. So dopamine is one type of the neurotransmitter that can be inhibited by cocaine. Okay. And another one is GABA. This one I just want to show you about the example of IPSP only, inhibitory supplementation process that I mentioned before. So the full name is gamma aminobutyric acid, GABA. So maybe in essay question, if you essay question or structure question ask you about the example of the IPSP, you can write this GABA. Full name is gamma aminobutyric acid. So it is a inhibitory neurotransmitter. So it inhibit the reaction one. So actually this is a mechanism, but actually you don't need to know it because it actually not our syllabus. But however, because it involve IPSP, so you should know it Okay. Okay, so I think that's all for today. Now it's already 9:30. So any questions so far? Or structure question ask you Okay, if no question, I will end the class today. So thank you for attending, and I will see you tomorrow again. Thank you.